Okay. So the basic idea is I'm just giving an intro and what are the things we have done already. The thing is, this is just to appreciate the fact that biomedical research is very time consuming and very, very, what do you call, competitive, as well as it needs a lot of investment in terms of skill, energy, as well as infrastructure and all other things. So what are the options for the researchers who belong to a resource limited setup? Okay. So consider that you belong to an this thing institute where resources are constrained. Okay, I give you a small example. Uh, Consider that you have isolated a plant extract which you think that would have very good bactericidal activity. Okay, or virucidal activity. So it will inhibit the growth of viruses and so on. Okay. Now, in order to do the bioassay to establish your what assumption that you have isolated a compound which is what this have some antiviral activity. So it needs a lot of investment in wet lab. Okay, how I tell you? First of all, you have to purify the product in the sense like if it is an alkaloid or things like that, then eventually you have to do HPLC and later on you can get a little extract. Now you have to deduce the structure of the extract or the alkaloid. So for that you need to do an infrared spectroscopy or some more sophisticated techniques. So finally you have deduced the structure. Okay. So now you will do the bioassay where you will culture the virus in cell, because viruses are what, as you know, intercellular parasites. You cannot culture them like this thing, what do you call, bacteria in a petri plate by giving some kind of medium. No, you cannot. You have to do a cell culture for this. Okay, which is very, very difficult to achieve. Okay, just once tell me, coordinator madam, whether I am audible.
Yes, sir, you're
And
audible.
if all our participants have already joined, then I'll move ahead.
Yeah, we
Yes,
can.
yes.
Everybody joined. Okay, fine. So what happened? So this was the point I was making, that if you are doing this particular wet lab study, so then what you have to need to do in the second step? In the second step, you have to culture the virus in a very controlled environment. You should have minimum a cell culture facility, eukaryotic cell culture facility. Then you have to also have a sufficient virology lab, at least biosafety 2 level of this thing lab. You must have a fluorescent microscope, and then all fluorescent dyes, tags, and all other things. Okay, still your job is not done, because a lot of trial and error you should do. You should identify the IMC, IC50 value, multiple different things. So what would happen if you have a simulation where you can predict the highest probability of any compound, that whether it will be virucidal or not. So I am not saying that you only entirely depend on the simulation. No, at least you can reduce the number of bad trials. Bad trials means a trial where you are getting wrong results. Okay, so consider that you needed to do at least 10 cell cultures. In the contrary, if you have a good simulation setup for you, you will end up doing only 2 or 3. Out of 2 or 3, at least one will be successful. So here what is happening? At least 7 bioassays or cell cultures where no need to be done, and therefore you can achieve very high economy or scale with a very minimum cost. Therefore, biomedical simulations are very important, and everybody should learn it. For example, somebody is from genetics, you should also learn it. Somebody from biomedical devices or things like that, they should also learn it.